Welcome to this Climate Lab tutorial. In this video, we'll be illustrating how to sample canopy, shrub, and herb cover at your study site. For this protocol, you'll need data sheets, a 50-meter measuring tape, a compass, a GPS, and ocular tubes. Proper use of the ocular tube is critical for the success of your sampling. The tube is designed to look at one very small section where the wires cross within the field of view of the tube. If green vegetation occurs directly behind this one spot, that is recorded as a 1. If there is no green, then record a 0. Here's another example of a 1 and a 0. Once at your study site, select a spot along your transect and run out your measuring tape 20 meters on each side of your transect. Use a compass to run the tape in a consistent direction. Make note of where along the transect you started and what directions you used for guidance, so that you can be consistent upon returning. Take a GPS location at the center point for future reference. You will collect data at each meter mark, totaling 40 points. At each meter, you will record a value for canopy, shrub, and herb layers. At each meter mark, look straight up with the ocular tube over a closed eye. Once in position, open your eye without moving the tube and determine if there's green vegetation behind the crosshairs. Repeat this process looking down. Any vegetation between your knees and your eyes is considered shrub, and any vegetation below the knee is considered herb. You do not need to identify what species of plant you're looking at, just whether it's present directly behind the crosshairs. We suggest sampling at 2 locations along one or more transects, collecting 80 points in all. It is important to collect data at these locations twice a year, once during the spring leaf out and again after leaf out, usually in the late spring. Please consult with Manomet staff to determine the best date ranges for sampling at your field site. By comparing the ratios of 1s to 0s, we can start to picture what the vegetation looks like and how it changes throughout the season. Over the course of many years, we may see differences in how the plant communities respond to warmer years and a changing climate.